Hi, my name is Caroline González-Vega, and I am a master student at the University of Illinois in the Stein Monogastric Nutrition Laboratory. And today it's my pleasure to talk about the determination of endogenous intestinal losses of calcium in canola meal fed to growing pigs. So this is the outline of this presentation. First, I would like to talk about endogenous losses, and then I will mention the objective of this experiment, the materials and methods that we used, also the results and the discussion, and finally some conclusions. This is a picture of the gastrointestinal tract of a pig to explain the definition of endogenous losses. So the green circle represents the nutrients in the feed, and the red stars represent the nutrients of endogenous origin that have been secreted into the gastrointestinal tract. Some of the nutrients that have been endogenous secreted are reabsorbed, but those nutrients that have not been digested and reabsorbed before excretion are considered endogenous losses. So the endogenous losses will be excreted with the nutrients from dietary origin that were not absorbed by the animal. So now let's see why endogenous losses are important to consider. For apparent total tract digestibility, abbreviated as ATTD, may not be additive in mixed diets if there is an endogenous loss of the nutrient, because ATTD values are influenced by endogenous losses. On the other hand, true total tract digestibility values, abbreviated as TTTD, are additive in mixed diets, but to calculate TTTD, it's necessary to correct apparent digestibility values for total endogenous losses. It has been demonstrated that there is endogenous losses of amino acids and phosphorus. However, there are not definitive data on whether or not there is endogenous losses of calcium from pigs. And also, there is no information about if adding phytase to the diets has an impact on endogenous losses of calcium. So the problem here is that we don't know if apparent digestibility values of calcium are accurate to use. But if in fact there is endogenous losses of calcium, apparent digestibility values are likely not additive, and therefore we expect that by correcting apparent digestibility values by the endogenous losses of calcium to calculate the true digestibility, these values will be additive and therefore will be more accurate to formulate mixed diets. So that is why we need more research in this area. So our hypothesis for this study was that endogenous calcium is lost from the gastrointestinal tract of growing pigs, and therefore values for true digestibility of calcium are different from values for apparent digestibility of calcium. And the objective was to determine the apparent digestibility and true digestibility of calcium in canola meal with and without added microbial phytase. For this experiment, we used 48 growing pigs with initial body weight of 16.7 kilograms, housed individually in metabolism cages. Also, the pigs were allotted to a randomized complete block design with 8 treatments, and we collect the fecal samples using the marker-to-marker approach. So we have 8 diets. All diets contain canola meal as an only source of calcium. 4 diets did not contain phytase, and these are represented by the orange color. The other 4 diets contain 1,500 units of phytase and are represented by the blue color. We use microbial phytase from E. coli from AB Vista, and this phytase is thermostable. In each group of diets, we have 4 levels of calcium, lower than the requirement to make sure we stay on the linear portion of the response curve. And these levels were 0.08, 0.16, 0.24, and 0.32. The reason that we used canola meal for this experiment was because we were looking for a plant ingredient that was high in calcium, and also in phytate. And we found that canola meal has 0.66% of calcium, which is pretty high for a plant ingredient, and also contains 2.58% of phytate. Also, we were looking for protein ingredients that we can use without adding more calcium to the diets, and we found that corn gluten meal and potato protein have 0.02 and 0.03% of calcium, respectively. So here we have a table with the composition of the diets. To increase the level of calcium in the diet, we increase the inclusion level of canola meal and reduce the amount of corn gluten meal, potato protein, and cornstarch and monosodium phosphate to maintain the protein and phosphorus level constant. We use monosodium phosphate as source of phosphorus instead of mono or dicalcium phosphate to avoid more inclusion of calcium. The other 4 diets were similar to these 4, but 0.03% of phytate was included at the expense of cornstarch. We calculate the apparent and true digestibility values of calcium of the diets. To estimate the endogenous losses of calcium, we use the regression procedure, and for this procedure, we regress back to 0 the calcium intake, and we analyze the data as a 4/2 factorial using ProgMix procedure in SAS. Now let's move into the results. First, let me set up the slide. In the x-axis, we have the level of calcium expressed as percentage. We have the 4 levels of calcium under the requirement, and these are 0.08, 0.16, 0.24, and 0.32. In the y-axis, we have the calcium intake in grams per day. The blue bars represent the canola meal diets without phytase, and the orange bars represent the canola meal diets with phytase. On the top, we have the p-values for the calcium level, phytase, and the interaction. The p-values on red are significant. So here we observe that calcium level was significant, which means that by increasing the level of calcium in the diet, calcium intake also increased, and phytase did not have an effect on calcium intake. For total fecal calcium output in grams per day, the p-values for calcium level, phytase, and the interaction between calcium and phytase were significant. And because we got a significant interaction, this means that we have 2 different slopes in the response, which means that the total fecal calcium output increased as calcium level in the diets increased. However, there is less calcium excreted when phytase is included in the diets, compared to the diets that phytase was not included. For apparent digestibility of calcium expressed as percentage, calcium level was significant, which means that by increasing the level of calcium, the apparent digestibility of calcium increased. And phytase was also significant, so the diets that contain phytase had greater apparent digestibility of calcium than the diets that did not contain phytase. And as I mentioned before, we use the regression procedure to determine if there was an endogenous loss of calcium. But first, let me set up the slide. In the x-axis, we have the dietary calcium intake in grams per kilogram dry matter. And in the y-axis, we have the apparent total tract digestibility of calcium in grams per kilogram dry matter intake. The orange line represents the canola diets without phytase, and the blue line represents the canola meal diets with phytase. By using the regression procedure, we obtain 2 linear equations. The negative intercept represents the endogenous losses of calcium expressed in grams per kilogram dry matter intake, and the slope represents the true digestibility of calcium. And we also got an R-square. So for the canola meal diets without phytase, we got a negative intercept of 0.16, and also we got a slope of 0.46. That means that the true total tract digestibility of calcium in canola meal is 46%. Regarding to the canola meal diets with phytase, we got a negative intercept of 0.19. Also, we got a slope of 0.70, which means that the true total tract digestibility of calcium in canola meal when phytase is added to the diets is 70%. So the two intercepts that we got from these two linear equations represent the endogenous losses of calcium, 0.16 for the canola meal diets without phytase and 0.19 for the canola meal diets with phytase. We test if these two values were different from each other, and they were not significantly different. Therefore, microbial phytase does not have an impact on endogenous losses of calcium in pigs. So once we determined that there was endogenous losses of calcium, we were able to correct the apparent digestibility values by the endogenous losses to calculate the true digestibility values of calcium. Remember before, the values of apparent digestibility of calcium change as we increase the calcium level, but it is not significant anymore with the true digestibility values. This is a similar concept that we have seen in phosphorus, and because of this, apparent digestibility of calcium are likely not additive, and we expect that true digestibility values are additive. The true digestibility of calcium was increased when phytase was added to the diets, from an average 48.2 to 68.8, close to the values obtained in the linear equations 46 and 70, respectively. And no significant interaction between calcium and phytase was observed. So with this, we conclude that endogenous calcium is lost from the gastrointestinal tract of growing pigs, and therefore, apparent digestibility values are different from true digestibility values for calcium. Also, we conclude that values for apparent digestibility of calcium are influenced by the level of dietary calcium, but that is not the case for values for true digestibility of calcium. And finally, we conclude that microbial phytase increases the apparent and true digestibility of calcium in canola meal, but it does not influence the endogenous losses of calcium. And with this, I want to thank you for your attention, and also we want to thank AB Vista for the financial support. And for more information about this topic and other topics related to monogastric nutrition, you can visit our website, nutrition.nsite.illinois.edu. Thank you once again.